Due on the 9th. That'll give me a few days to be able to grade them.
Have you graded the module?
The 9th?
Mhm.
And do you have a
I think 11:59. No, that doesn't mean that you can't turn it in earlier.
Yeah. I didn't know that. I don't know why I thought it would be on.
No, the 14th is the final. Because I'm requiring this of everybody, I have to have time to grade these. Like it just comes down to time management on my part. Right. Yeah. So everything for this module, I think, should be due on the 12th, and then the final is due on the 14th. This is different though, just because once again, I have to have time. There's just simply no way I can grade all of these papers and all of the module 4 exams in 3 days or 2 days, much less than having to grade the finals. So I just need a little bit more time than that. No. So all this is this is a, like, it is a full lab report. So it's going to have kind of the 5 major sections of a scientific paper. So we're going to have the introduction. We're going to have a materials and methods. We have a results, a conclusion, and then the literature cited page. Okay. So the reason I say 2 or 3 people here is because one person can do the intro. One person does the methods and the results. One person does the conclusion. Then you can have everybody work together on the literature cited. That's because the introduction and the conclusion are both going to cite things, like they're going to have in-text citations, at which point they just include that information in the literature cited page. And then they just need to kind of put everything in alphabetical order, and then we're done. Okay. It's fairly straightforward. It's going to basically work the same way as last time. So last semester, y'all got a little bit of practice writing the introduction. The discussion or the conclusion is basically the same thing as your introduction, but in reverse. Okay. But that's pretty much it. We have the same technical guidelines. So we have the same kind of font stuff, which is just 12-point Calibri or something that looks like Calibri. When I put Calibri on here, it's because this is a Mac thing. And then I realized that Windows machines don't necessarily have it. But basically pick a font that's easy to read, not like, I don't know, one of the weird ones. I know there's a bunch of weird ones. But Calibri, Arial, Calibri or Arial is fine. It is going to be, wait, hold on. Did I include? Oh, snap. I need to fix that. This is not extra credit. This is just 10 percentage points. So it's worth 10 points. I need apparently to fix that. This is leftover, or that line was left over when it used to be extra credit. All right. We have to have citations. So every time that you say something in either the introduction, the materials and methods, or the results, or the conclusion, that would not be considered common knowledge, you need to cite that source. Okay. You'll be citing your book, which is, no, okay, that's our lab manual. Cite your book, find.org, cite that. Or you can also just simply go on to Google Scholar. So if you kind of type up here, Scholar, Google Scholar, go on to Google Scholar and then type, say, what do we want to look for? Type urinalysis review. Hey, here we have a paper right here, published in 2005. That's close enough. Oh, here's an even better one, 2015. Actually, we click that one instead. No, don't load it. Especially if you type review, this will basically pull up a page or pull up an article that will have sometimes hundreds of citations. And when it came to writing, a review article is very similar to something like a textbook chapter. It's going to contain a lot of information from a lot of different places, all boiled down. Okay. So these are great as like a very first thing to read, like when you're trying to read about something. These are also a really great place to go to find additional sources. Okay. Because if I look in here, like say I start reading this, it's going to have tons and tons and tons of citations just mixed through all of these. Okay. This is actually a really nice little article here. So this might be a great one to cite. So here's one here. You can sometimes download PDFs. I basically collect PDFs on my computer. It's kind of ridiculous. But find this when you look at its literature cited page. Look at all these. Look at them. Like many of these you might find useful. So read through some of these titles and see if it helps you. Hey, dipstick urinalysis and the accuracy of the clinical diagnosis of urinary tract infection. Hey, you could use that one if you're talking about urinary tract infections. When you're reading these, also in their introductions and conclusions, they might give you some idea of kind of what to talk about, how to talk about it, that sort of stuff. Okay. So there's a lot of information to be found out here. So this one, I would even just type this now. Type urinalysis, a comprehensive review, Simmerville et al. There you go.
Would we just cite this, or would we have to go all the way down on where it actually comes from?
Oh, you can cite this. This is fine.
Okay.
Because like your textbook, if you actually look at the back of every chapter in your textbook, it will actually have citations, like where they got some of the information that they put into the textbook.
Okay, so we're citing like a review, we would have to go find who actually wrote
No,
it.
no. But you can use their review and you can still use some of their sources. So if you are struggling to find more sources for things, like you just didn't want to keep citing this person over and over and over again, you can go down into their literature cited and find a couple sources there, and you can use that to kind of increase your number of sources. Okay. I will say this is probably one of the better ones of these I found. Okay. So this would be a good one if y'all want to take a picture of this one just so you can have that title there. Very, very, very helpful. Oh, where's the cursor? There we go. All right. So that's basically how I would write the report. Once again, this is going to be submitted through SafeAssign as well as AI checkers, so don't do that.
It's for a guide on how to do it.
Yes, I've even kind of drawn it right here. So this is basically the simplest version of it. So you do the last name, the initials of the first author, second author, last name, initials, so on. The year it's published. So in this case, it's 2019. So Marieb, EN, Smith, LA, and Zal, PZ, published in 2019. And then the title of the article. So in this case, it's the title of the book. So Human Anatomy and Physiology Laboratory Manual, and the publisher of this book was Pearson. If this was, say, this paper right here, it would be Simmerville.ja, comma, Maxted.wc, comma, and Pariah, comma, J.J. And this was published in 2005. So parentheses 2005. It's called Urinalysis, a Complete Review. So that would be italicized. Here, let's write it out. Simmerville. It's like one maximum. Next. Let's see. Five, and then italicized because it's the name of the article. Comprehensive review. I'm underlining because I can't italicize the board. And then this is published in no, American Family Physician. Okay, here we go. So here we go. Don't do that. They do it wrong. They almost always do it wrong. Like literally every single time they do it wrong.
Use citation.
Yeah. Yeah, if you use a citation manager, they are more likely to get it right. Even sometimes they'll still not quite get it right. You actually still need to go read through it.
Yeah, I wasn't actually sure.
Yeah, that's part of the problem too, because you have to know what each of these parts are. So this is the journal. That's what this American Family Physician is. This is the journal, and then the edition of the journal is number 71, issue 8. So 71, 6. And we're done. You can also include the page numbers. I think technically it's supposed to include the page number.
And then in the text, we would put all those names, which was five.
Yep. Oh, no, so this is your indolent cited. Anything more than 2, you can just use at all. So in text, it would look like this. It would just be Somerville et al. 2019. That's what it would look like in text. Does that make sense?
I didn't know that all that, and whatever it would say that in text.
It just means and others.
Okay.
Stupid Latin. But yeah, just means and others, and then you do the year. And that way, whenever someone's reading something, they can say like, oh, okay, Somerville, like, then when they want to check your citation, so you said this in your in-text citation, then when they want to check this, they'll go into the lit cited and see the rest of this, and then they can go get it. Or you cited it here. This says, hey, I'm going to include the rest of this citation information if you are curious in the literature cited page. So you have to have it listed in the lit cited page. If it's in your lit cited, it needs to be at one of your in-text citations somewhere in the paper. Same thing goes if you use it as an in-text citation somewhere in your paper, you have to include that information in the literature cited. They have to be meshed. Does that make sense? All right, so that's it as far as citations. Let's go ahead and do this part. There's also a link right here that goes into how to do that too, like helping you with formatting. So if you're using like a website or something like that, it will help you format some of those things. We talked about writing it a little bit. The report specifics. So this is basically, like I said, about the urinalysis lab, basically the one that we just did. Y'all only have 2 data points. So if you wanted to exchange data with one another, you can use you as subject 1, subject 2, whatever. You still need a couple more. That's where you're going to still need to do the urinalysis lab online. But now hopefully that lab makes a little bit more sense. I don't know if y'all have done that ahead of time or not, but hopefully now we can put that into a little bit of perspective. Essentially what you're going to do here in the introduction is you're going to talk about kind of overall point of the urinary system, like what it's trying to do, what it's trying to regulate, and then you can talk about some of the things that can go wrong. Okay. So first paragraph, talk about normal systems. Second paragraph, the type of things that you would expect to be found in the urine, why you might expect to find these. Like, hey, like we are Americans, especially Americans with high protein diets, you would expect to find ketones in the diet. But then you can also start in the third paragraph, talk about the things that you shouldn't find there. You shouldn't find urine, you shouldn't find glucose, and then talk about why you wouldn't find those things, why it would be important for you to find out if those are present in someone's urine and what that tells you about their, not just their health and their kidneys, but their overall health. Like glucose being found in the urine tells us, hey, we either have major kidney problems or we have diabetes. Both of those are going to kind of affect overall health of the organism, of the person, not just that system, right? So you can kind of use those as talking points, okay? Then in materials and methods, this is basically where you're going to talk about what we did. So in this case, you collect a sample. Now, how you collect those samples could actually be important. I didn't tell y'all any particularly systemic way to do it. However, that urinalysis, a complete review, would actually tell you the best way to go about doing it. And you can kind of use that to see if you did it the right way or not. Basically, what it's going to say to do is like start to pee a little bit. That way you kind of flush anything that may have creeped into the opening of the urethra out. And then you finish peeing into the cup. That's essentially what they're going to tell you to do there. Just that way you can, like I said, mitigate anything that you don't want it to act like you have a urinary tract infection if you actually don't. Then we can move down from there and go into how we actually tested. In this case, we tested with these particular urinalysis test strips. If it were me, I would write down even the name of the urinalysis test strip, as well as all the things that it's testing for. Okay, so these urinalysis, these are 10 test strips. These test for leukocytes, nitrites, urobilinated protein, blah, 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 blah. Okay. And then in the results section, you're just going to list what everybody had. Okay. So for yourself and then the other couple of people from the urinalysis experiment or in the virtual lab, you're going to say, hey, this person was negative for leukocytes, negative for nitrites, negative for protein, pH of this, blood of the, or negative for blood, negative for ketones, negative for bilirubin, negative for glucose, a specific gravity of X, and then basically do that for everybody. Now, if a number is, say, really high, so say someone's glucose was, see, what was one of the ranges on glucose? Like say, like one to mid to high level ones on glucose. 500. Okay. So say instead of someone having 0, they had 500. Well, 500 watts, nanograms per microliter. Milligrams per microliter.
Oh. Yeah.
Milligrams per deciliter. Okay. So basically units. Well, that is, well, infinitely higher than what would be considered normal, right? So a normal range, like say you even give it to say 1, that is 500 times more than normal. That is a problem. Okay, so don't just simply say like, okay, they have this number of 500. You could like in the results section, you can say this is 500 times more than the normal amount. That tells us something. Okay. That by doing those types of comparisons, it makes it easier for people to kind of understand what is going on. Okay. We are like, our brains tend to work in terms of proportions. So doing that type of, it's not really math, it's truly just like, hey, this type of comparison can just make it a little bit more obvious to the reader what's happening and why that may be important. Then in the discussion, then you're going to talk about like, okay, this person had all normal values on these things, which means they're probably a perfectly healthy person. This person had 500 nanograms per microliter of sugar in there. Well, what does that tell us? This tells us this person may have diabetes. They probably need to follow their doctor. What are some of the implications if they had diabetes, right? Like, what does that tell us? Why does someone having diabetes lead to this? You can maybe talk, like, give a sentence to explain like how someone having diabetes would lead to having urine or sugar in their urine. And you're basically going to do that for each subject. And then you can kind of circle back around to, hey, urinalysis is a useful experiment to look at not just urinary system health, but also like health as the whole. And then you're done. That's basically kind of your flow through for the experiment. Does that make sense? This is not a whole lot. This should probably be 4 pages, 5 pages, something like that, even including the literature cited page. So this isn't a crazy amount or anything like that. As far as citations, I think I'm asking for 3. There's 1 right there. You have your book, that's 2. So basically you have a free slot. But however, if you go to that page that we found, you can find dozens more. In Google Scholar, like I said, you can find dozens more. So it's pretty easy to find these types of things, and they're usually pretty useful. Does that make sense though? Kind of makes sense what our goals are. All right, so y'all have any questions then about the urinary system?
How did you know this is? What chapter?
This is 24?
No.
Yep, 24. And there's really not a whole lot of anatomy to talk about for chapter 25. Chapter 25 is honestly just chapter 24.2. That's basically what it is. It's just more information about kind of the types of things that what the kidneys are doing and the types of things that they are regulating and how. So unfortunately, not a lot of anatomy. That's just all kind of physiology stuff. So not a lot of lab stuff. No. Yep. Although, like, we used to do the urinalysis experiment where we would have people drink things. I just forgot to bring stuff for y'all to drink. Where we would have one person drink, say, like sugar water, and another person just drink like not dialysis water. It's essentially just bottled water, but even pure bottled water, like where there's nothing dissolved in it whatsoever. And we could actually look at the differences and stuff that would cause like downstream. But there's 2 of y'all. So that makes it not necessarily all that useful. but that's pretty much it. Like this is fairly straightforward. This should be pretty easy. At this point, y'all have written a couple of introductions. I think you maybe even did, I think you even said it just now that y'all like wrote the entire report rather than just the introduction. So you should be fairly well practiced at this point.
Wasn't it great?
Huh? Hey, it's still something. So no questions? All right. So you can go ahead and just take your urine cups back to the bathroom, pour those down the toilet, flush it, rinse them out, and then you can just bring them back in here and I'll get them clean. I'll go get a bucket to put them in. The test strips can just go into the regular room. Well, actually, let's put those in here just in case. Mackenzie, any questions from you? Awesome. Well, if you do develop any or come up with anything, just let me know.